Hello and welcome back. If you have not subscribed to this channel, please go ahead and click the subscribe button. Today we'll learn about some biochemical methods used for identification and differentiation of bacteria. There are 5 I's of basic microbiology lab procedures that are used to examine and characterize microbes. In my previous videos, we have covered 4 of these that include inoculation, incubation, isolation and inspection, both macroscopic and microscopic. In today's video, we will cover 5th component of the 5 I's, that is identification. The biochemical tests are a very important part of the identification process. They shorten the time required to identify microbes, reduce the costs, and enhance the accuracy of identification of a patient sample. Various types of biochemical tests that are in use include single enzyme tests like Catalase, Coagulase, Oxidase, Urease, Indole, PYR test and Hippurate hydrolysis. Amino acid degradation test, for example, lysine, ornithine, arginine decarboxylation test, deamination test. Single substrate utilization tests like citrate, malonate or acetate utilization. And finally, the tests that establish inhibitor profiles in presence of salt, surfactants, antibiotics and toxins. The bacteria were born 3.8 billion years back when there was no oxygen in the environment. The bacterial metabolism was entirely dependent on enzymes that had iron-based cofactors. Iron was abundant on the Earth's crust and bacteria thrived in anoxic environment. Later, photosynthesis began and oxygen got accumulated in the environment. Oxygen entered the bacterial cells and oxidized the iron-based cofactors, inactivating their enzymes. In this process, the bacteria were killed. More and more bacteria died because of oxygen toxicity, and the only place where bacteria could survive was where there was anoxic or anaerobic environment. Over time, some bacteria adapted to the environmental oxygen by changing to metabolic pathways that were not dependent heavily on iron-based cofactors. Oxygen could still enter and harm aerobic bacteria by stealing the electrons and inactivating the essential enzymes. Single electron transfer produces superoxide and transfer of 2 electron produces hydrogen peroxidase. These are more potent oxidants than oxygen itself and can bind to any iron available inside the cell, thus producing hydroxyl radicals. These oxidants and hydroxyl radicals can easily kill a bacteria if allowed to accumulate. As part of their evolution, the aerobic bacteria developed special enzymes that could degrade these oxidative molecules. Examples of these enzymes include catalase and superoxide dismutase. Catalase enzyme takes care of hydrogen peroxide by degrading it to water and oxygen, and superoxide dismutase takes care of superoxide. These adaptations helped some bacteria to grow in the presence of oxygen and they became aerobic bacteria. The bacteria that persisted with iron-based cofactors like their forebearers are now called anaerobic bacteria and can survive only in the absence of oxygen. Now let us study the catalase test that detects the presence of catalase enzyme in the bacteria. Catalase positive bacteria can respire using oxygen as a terminal electron acceptor. They include obligate aerobes and some facultative anaerobes. Some examples are Staphylococcus species, Micrococcus species, Bacillus, Propionibacterium acne, Helicobacter pylori, Haemophilus influenzae, Moraxella species, Neisseria species, Pastorella species, and Brucella. The catalase negative bacteria do not respire using oxygen as the terminal electron acceptor. They include obligate anaerobes or facultative anaerobes. These bacteria may have other enzymes that catalyze hydrogen peroxide and other oxygen radicals. Examples include Streptococcus species, Enterococcus, Clostridium, Haemophilus ducreyi, Echinella corridens, and Kingella species. Items needed to perform a catalase test are clean glass slides, wooden applicator. Do not use iron-based metal loops that will react with hydrogen peroxide. 3% hydrogen peroxide stored in brown bottle under refrigeration. Fresh culture plate with well-isolated colonies, 18 to 24 hours incubation. Older cultures are not used because they may contain the non-viable or dead bacteria and can give false negative catalase reaction. Positive control, Staphylococcus aureus. Negative control, Streptococcus pyogenes. Transfer a small amount of bacterial colony to the center of a dry and clean glass slide using a wooden applicator, and the colony should be visible with the naked eye. Put a drop of 3% hydrogen peroxide onto the colony. Mix them well using the wooden applicator. The rapid and sustained appearance of bubbles or effervescence within 5 to 10 seconds constitutes a positive test. In a catalase negative organism, there will be no bubbles or only a few scattered bubbles. Because some bacteria possess enzymes other than catalase that can decompose hydrogen peroxide, a few tiny bubbles forming after 20 to 30 seconds is not considered a positive test. Do not use a metal loop or needle with hydrogen peroxide. It may give a false positive catalase and can degrade the metal. When using colonies from the blood agar, be careful not to pick media containing the RBCs as they contain catalase and can give false positive catalase reaction. Some clinically important uses of catalase include differentiating staphylococci that are catalase positive from streptococci and enterococci that are catalase negative. A semi-quantitative catalase test is used to identify Mycobacterium tuberculosis. It can be used as an aid to the identification of Enterobacteriaceae family that is catalase positive. Now moving on to the coagulase enzyme that is produced by pathogenic Staphylococcus strains. This enzyme converts fibrinogen into fibrin threads that form a mesh around these bacteria, which protects them from antibodies and phagocytosis. The presence of coagulase differentiates pathogenic Staphylococcus from non-pathogenic Staphylococcus strains that do not produce coagulase. We can detect the presence of coagulase enzyme by two different methods. First is slide coagulase test and the second is tube coagulase test. Slide coagulase test is done to detect the presence of bound form of coagulase enzyme, also known as clumping factor. Clumping factor is a bacterial cell wall receptor that binds fibrinogen. So let us see what happens when we suspend the bacterial cells in the plasma that contain fibrinogen. The clumping factor binds and converts the fibrinogen in the suspension into elongated fibrin strands. The fibrin strands form a meshwork around the staphylococci. The meshwork entangles the suspended bacteria, causing them to clump together. This can be seen as visible clumps that are diagnostic for the presence of bound coagulase or the clumping factor. The sensitivity of the slide coagulase test is low. It is not produced by all pathogenic Staphylococcus aureus strains and may give false negative results. So the negative slide coagulase test must be confirmed by doing the tube coagulase test, that is a more sensitive test. Free coagulase is produced by Staphylococcus and can be detected by doing the tube coagulase test. The free coagulase is also called staphylocoagulase. It reacts with coagulase reacting factor, that is a thrombin-like substance. This combination of coagulase and coagulase reacting factor converts fibrinogen into fibrin strands in the solution. This leads to the formation of a visible clot. Items needed to perform a coagulase test include clean glass slides, test tube, wax pencil, wooden applicator sticks, fresh culture plate with well-isolated colonies, rabbit plasma with EDTA. Do not use citrated blood. It can give false negative results. Control positive strains of Staphylococcus aureus and control negative strains of Staphylococcus epidermidis. First, we take a clean, dust and grease-free slide and make a small circle in the center of the slide with a wax pencil. Put a drop of distilled water or normal saline onto the center of the slide. Now pick a small portion of the colony and transfer it to the drop of water on the slide. Gently emulsify the colony material from the organism to be identified in the liquid with minimal spreading. The isolate should form a smooth milky suspension. Now place a drop of undiluted plasma in the suspension. Mix with a wooden applicator stick. Make similar suspensions of control positive and control negative strains to confirm the reactivity of plasma. Observe for agglutination of the test suspension. A positive reaction will be detected within 10 to 15 seconds of mixing the plasma in the form of white precipitate formed by the coagulated bacterial cells. The test is considered negative if no precipitates are formed within 10 to 15 seconds. The slow reacting strains should be tested with the tube coagulase test. The negative control should remain smooth and milky white. If the control suspension agglutinates as well, then the test is uninterpretable. All strains that are coagulase positive can be reported as coagulase positive Staphylococcus, or if the colony morphology is appropriate, as Staphylococcus aureus. All strains producing negative slide test must be tested with the tube coagulase test. Now coming to the tube test for free coagulase. First, we prepare a 1:6 dilution of the plasma in saline and put 1 ml of the plasma in small test tubes. Then emulsify several isolated colonies of the test organism in 1 ml of plasma. It should form a milky suspension. Now incubate the tube at 35 degrees centigrade for 4 hours. Examine after 1, 2 and 4 hours for clot formation. Observe by gently tilting the tube for evidence of clotting. The tube should be gently tilted and not agitated because this may disrupt the partially formed clot. The tube coagulase test is considered positive if any degree of clotting is noted. The clot in this example is evident as the plasma has become solidified and is not flowing out of the tube because of clot formation. The tube coagulase test is considered positive even if a small amount of clot is noted. If no clot is observed and plasma flows on tilting the tube, keep the tube at room temperature and read again after 18 hours. Some examples of reporting done using coagulase and catalase tests are as follows. Tube coagulase positive, catalase positive, and gram-positive cocci in clusters. The diagnosis is confirmed as Staphylococcus aureus. Tube coagulase negative, catalase positive, gram-positive cocci in clusters, creamy white colonies. The diagnosis is coagulase negative Staphylococcus. Slide coagulase positive, beta hemolytic colonies are diagnostic of Staphylococcus aureus. Slide coagulase test positive and non-hemolytic colonies on blood agar. This combination is non-diagnostic, so go for tube coagulase. If slide coagulase test is negative, go for the tube coagulase test. So that was all about the coagulase test. Now we proceed to the next, that is oxidase test. Some aerobic bacteria can utilize oxygen as the terminal oxygen acceptor in the respiration. They do so by having cytochrome C oxidase as the terminal enzyme in the respiratory chain. This enzyme reduces oxygen into water and produces ATP in the process. We can detect presence of this enzyme in the bacteria with the help of oxidase test. Not all aerobic bacteria have cytochrome C oxidase, and they can still respire in oxygen using other oxidases in the electron transport chain. All anaerobes will, however, be oxidase negative. This test helps us differentiate the Enterobacteriaceae family, that is oxidase negative, from oxidase positive Pseudomonas species, Aeromonas species, Vibrio, Campylobacter, Pasteurella and Neisseria species. Items needed to perform the oxidase test include filter paper, disposable plastic loop, fresh colony of test organism or nutrient agar, 1% Gordon or MacLeod reagent also called oxidase reagent, positive control that is Pseudomonas aeruginosa and negative control that is Escherichia coli. First I will describe the filter paper test method. In this we soak a small piece of filter paper in oxidase reagent. With a sterile plastic loop, pick up isolated colony from a young culture plate and rub it onto the treated portion of the filter paper. Do not use nickel, steel or other wire loops. Oxidase positive organisms will change the color to dark purple in 5 to 10 seconds. Oxidase negative will show no color change even after 2 minutes. And delayed oxidase positive organisms will show color change after 10 seconds but within 90 seconds. Another method is filter paper spot method test. In this we use a loop to pick up an isolated colony from the young culture and rub it onto a small piece of filter paper. Now add 1 to 2 drops of oxidase reagent on the colony smeared on the paper. Oxidase positive organism will change the color to dark purple in 5 to 10 seconds, whereas oxidase negative will show no color change even after 2 minutes. Another method is the direct plate method, in which we directly place 1 to 2 drops of oxidase reagent onto the well isolated colony of bacteria on a nutrient agar plate. Observe for the color changes. The interpretation is similar to that of the filter paper method described above, where oxidase positive organisms will change the color to dark purple in 5 to 10 seconds and oxidase negative will show no color change. So that was all about catalase, coagulase and oxidase test in this video. Hope you like it. Please like, subscribe, share and comment. Thank you for watching.